Okay, ladies and gentlemen, so today we're actually going to do a little lab activity. So you're going to find this packet and then a packet of pieces in your green folder. And we're starting with this handout first. And as we go through this handout, basically we're doing a cut and paste, and we're going to be making strands of DNA. And this is only an example. So please, please, please do not make it exactly like this. In the bigger packet that you have 4 pieces and they have different shapes. So these are the nitrogen bases. And there are 4 of them for DNA, A and T and C and G. And you can see like they're pieces of a puzzle, so they will fit together. And then actually each of these, the little top part is the phosphate group and the big part is the sugar. So the sugar would be deoxyribose sugar. And then the little part at the top here is representing a phosphate group. So each one of these is representing a nucleotide. So we have deoxyribose sugar, a phosphate group, and a nitrogen base. So these 3 parts make up one nucleotide. So you're looking at 4 nucleotides there. And then if you go on, it asks you to cut, but I'm going to suggest you not do the cutting first. I'm going to say, let's start here at number 5, and then we'll work our way back to number 4, and then we will continue on and on and on from there. Things you do need are going to be to get some colored pencils or markers or whatever you have. A is going to be represented by red. T, thymine, is going to be represented by blue. C, cytosine, will be represented by green. And then G, guanine, will be represented by yellow. So if you have those 4 colored pencils available, then you can get them out. And we're going to go to this packet that has all of our pieces in it. So I've previously colored some of these, so we're not taking forever to do it. But in this packet, you will notice that there's 4 pages that are exactly the same. And you're going to take your red colored pencil, and then you are just coloring the nitrogen base. So here, the A is representing the nitrogen base. These will be what we call the rungs of the ladder, the part you step on. The top part of this will be a phosphate group. Ooh, phosphate. The P group. And then the sugar. In this case, it's deoxyribose sugar. So you're going to take your red pencil and you're coloring just the nitrogenous base in each of the 4 pages red. So make sure you have 4 of them, and it is just the A's that are red in color. Then you'll grab your blue, and T, the thymine, is blue, so you'll be coloring your thymine, just this part blue. It doesn't have to be perfect coloring, so don't stress over that. We're going to be cutting them out, so anything on the outside edges will get cut away anyway. Again, go through and do all 4 in blue. The C will be green. So you're coloring your green C's all the way in the packet, all 4, and G will be in yellow. So once you have all of your pieces colored, so it would be 16 pieces, 4 adenines, 4 thymines, 4 cytosines, and 4 guanines, we're going to take the whole little packet and cut them out. My suggestion is you hold it up to a light or a window or something so that you can see that they kind of match, and it will save you some cutting time. We are cutting out all of the pieces. So once you get the pieces cut out, you are going to be arranging them in one way. So hopefully if you line them up pretty good, you'll be able to get all 16 of your pieces cut fairly quickly. If you want to cut them one by one, you certainly can do that. And then the next step after this, if you read the directions of the lab, is going to be to put them together in some way so that they will match. What you're going to notice when you do this is that some of the pieces are going to be in reverse to each other. So let's leave you for that for right now. And this will be the setup of this lab. When we come back, we're gonna have all our pieces cut out and we're gonna show you how to put them together. So we'll see you in a little bit, get the coloring and cutting done. Thank you.